Hi everyone, I welcome you all to this online lecture on the titration of amino acids. And today we will be understanding the titration of amino acid. But before starting the lecture, we need to understand certain physical properties of amino acids that are required so as to understand the titration of amino acids. So we know that at physiological pH or at around the neutral pH, the amino acids, it exists as a zwitterion. Now, what this zwitterion means, this zwitterion here you can see it is carrying a positive charge as well as a negative charge. All right. So now this is at neutral pH round about 7. All right. But when the pH is decreased, when the pH is decreased, that means there are more and more protons outside the system. When there are more and more protons outside the system, you can see here the COO negative group, it has changed to COOH group. That means it has accepted the proton. It has accepted the proton. And when you can see, let's say if this reaction is reversible, now when you will increase the pH above 7, let's say it's 8, it's 9, it's 10. All right, there are certain amino acids that are completely neutralized at about pH 9. All right. So when you increase the pH, when you increase the pH, then in the system there is decreased hydrogen ion concentration. When there is decreased hydrogen ion concentration, here from this NH3 positive, one H will be removed. One H will be removed, minus H. So it becomes an anion. Here you can see it is negatively charged. So we need to understand this fact that in water, whenever the amino acids, they are being dissolved in water, they can act both acids and bases. Now, what are acids? Acids are proton donor. Acids can donate the proton, all right, and bases can accept the proton. So yes, these amino acids, they can behave both as acids and as base. So let's talk more about these zwitterions. Zwitterions have one positive and one negative charge. We already discussed, and hence they are electrically neutral. I told you that whenever an electric field will be applied, so they since they are carrying no charge, the net charge is zero. All right, net charge is zero. When the net charge is zero, the amino acid will not move when uh, 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 electric field will be applied. So they are electrically neutral. And the pH, the physiological pH at which, see, different amino acids have different pi. Okay, what is pi? Pi is the isoelectric point, and this pi is very much important whenever we need to understand the titration of amino acids. So, what is isoelectric pH? The isoelectric pH is that pH at which the amino acid carry no net charge. There is no net charge. Net charge will be 0. All right. For example, the isoelectric point for glycine is pH 6.0, which clearly indicates that this glycine amino acid will be in its Zwitter ionic form at pH 6.0. So here you can see when the amino acid is containing positive as well as negative charge, it is in its zwitterion form, all right. But when pH is acidic, that means in acidic pH, more and more protons will be in the system. When more and more protons will be in the system, then what will happen? Here you can see this carboxyl group, it has acquired one H. And when the pH will be basic, when the pH will be basic, then that means there will be more OH ions in the system. And when there will be more OH ion in the system, then what will happen? then the proton will be removed. So in these case, when the pH is acidic, the net charge on this amino acid is +1, and when the pH is basic, the net charge is -1, and net charge is 0 at about neutral pH. So this pH is known as isoelectric point. So now we need to understand one important fact, that what is pH? pH is negative logarithm of hydrogen ion concentration. Now if this pH is less than Pi, then net charge will be +1. Similarly here in this image, if the pH is less than, here the pH was less than Pi, isoelectric point, the net charge will be +1. That means then the amino acid will be acting as what? It will be acting as a proton acceptor. Now, when the pH is equal to Pi, net charge is 0. When the pH is equal to Pi, then the amino acid will be stable as a Zwitter ion. And if pH is greater than Pi, then the net charge will be -1, and the amino acid will be acting as a proton donor, increasing the concentration of OH. Then what is happening? That deprotonation is happening. Deprotonation is going on. Now, from where the deprotonation is going on? If we proceed from left to right, that means we are increasing the pH. Increasing the pH. When we are increasing the pH, we are increasing the concentration of OH ions. And when we are increasing the concentration of OH ions, deprotonation is going on. So first of all, the deprotonation will start from the carboxylic acid. All right. So now the carboxylic acid COOH, it is being dissociated into COO negative and H positive. All right, and H2. All right. So when this COOH is being dissociated, then what is happening? Please understand what is happening. You can see that the dissociation constant pK1 is the dissociation constant for this carboxyl group. Now, when more and more pH is being increased, that means the OH concentration is increasing. Then, secondly, the deprotonation of amino group will be there. When the deprotonation of amino group will be there, the proton will be removed from the amino group. All right, the proton will be removed from amino group, and the dissociation constant pK2 is for the amino group. When we need to understand the titration of amino acids, these are the basic things which we need to understand, that whenever we are increasing the pH, there is pK1. This is the dissociation constant of COOH, and pK2. This is the dissociation constant of amino group. All right. Okay. So let us move on to the this is the titration curve for glycine. This is the titration curve for glycine. Now, from this glycine amino acid, we can see clearly that this is existing as a Zwitter ion. This is existing as a Zwitter ion. When this is existing at a Zwitter ion in a pH, at a pH, and that is the isoelectric point Pi. That is the Pi. Now the main question is that how to calculate the Pi. How to calculate the Pi. See, these titrations, they are acid-base titrations. Whenever we need to titrate any acid, we just titrate it with a base. So here you can see that here the glycine amino acid, it is in protonated form. It is in protonated form, and gradually the pH is being increased. Gradually the pH is being increased, and we see that as the OH equivalents are being increased, the pH is being increased, and the amino acid loses its proton. The amino acid loses its proton. And when it loses its proton, it loses its proton at a pH a pH, and the dissociation constant pK1, it is 2.34. It is 2.34. This is 2.34. And when further increase in pH is going to happen, then it is neutralized. The amino group, here you can see the amino group is also and it is also losing a proton. When it will be losing a proton, the pK2, the dissociation constant for amino group, pK2, this is 9.60. And from here, we can calculate the pI for glycine amino acid. What is the pI? The isoelectric pH, at which pH the glycine, the structure of the glycine will be a zwitterion. So how we can calculate? We calculate this pK1 plus pK2 divided by 2. So it comes out to be 2.34 plus 9.60 divided by 2. So it comes out to be 4, 6 and 3, 9, 9 to 11, 11 upon 2. So it comes out to be 2 5s are 10, 5.97. So this 5.97 is the PI for the glycine. So this we need to calculate. Okay. I'll also tell you that what is the need of calculating these PIs. So let us now consider the, amino acid glutamate and its titration. All right. So now understand that the glutamate amino acid, this amino acid is a triprotic. All right. We are getting 3 pKa values. We are getting 3 pK values for this glutamate amino acid. So this is the titration curve for glutamate amino acid. We will be, what we are doing, we are increasing the concentration of OH negative ions. All right, we are increasing the pH. Also, we are increasing the pH. As we are increasing the pH, what is happening here? You can see that there are, at the amino group, there is a positive charge. There is a positive charge at the amino group. All right. And when there is a positive charge in the amino group and we increase the pH, that means we are increasing the concentration of OH negative ions as we are increasing the pH. Then what happens? There will be deprotonation. And here you can see the deprotonation is of carboxylic acid. The deprotonation is of carboxylic acid. And here forms the zwitterion. This is the zwitterion. Why this is the zwitterion? Because here the negative and positive charges are there. Here the negative and positive charges are there in the alpha carbon atom. Since the R group of the amino acid, where is the R group here? Here is the R group. Since the R group of amino acid is still protonated. So when we will further increase the pH, what will be happening? When we will be increasing the pH, the, you know, when we will be increasing the pH further, what will happen? This C carboxylic group of the R group, it will also get deprotonated and the amino acid will carry a charge of -1. Now, in further increasing the pH, what will happen? This amino group, which was still, this amino group was still protonated, this will also lose its proton and will attain a charge of -2. Now, question comes that when there are, there are three pKa values, which value to take for calculating the pi? Now, here, what we need to understand clearly that the values which is changing the structure of Zwitter ion, only they need to be taken into consideration. हम यहाँ पर pK2 की वैल्यू नहीं लेंगे क्योंकि Zwitter ion के साथ में कौन सी वैल्यू है, pK1 और pKr. तो आप यहाँ पर देखेंगे, ठीक है, the dissociation, the pK1 was, what was the pK1? pK1 value was 2.19 and pKr value was 4.28. We will have to take these two values for calculating the pi. When we will take, see, 2.19 plus 4.18 divided by 2, then the pi comes out to be 3.22. If you will calculate, you will get the value 3.22. That means we don't have to take all the pKa values, all the dissociation constant values. नहीं, हम सिर्फ वो दो values लेंगे जो Zwitter ion के आसपास होंगी. So this is how we calculate the pi values, and this are important and hold the significance in examinations, in various examinations, and also in our body application. Jitni bhi application hogi, they will be related to this pi only, because at pi the amino acid, it will be existing as a zwitterine structure, then only it will be acting as a buffer. Okay, now again, this is histidine, this is histidine. So here in the histidine example, you can see that it is existing as a Zwitter ion here. All right. It is existing at a Zwitter ion somewhere here. Now, when the pH is being increased, when the pH is being increased regularly, okay, you can see the structure of histidine from acidic pH. We are increasing the pH here. When the pH is acidic, what is going to happen? It is attaining a charge of +2. Gradually, when the OH ion concentration will be increased, deprotonation is going on. Where is the deprotonation going on? The proton is being removed from this carboxylic, okay, this carboxylic group. Then again, the pH is increased, and now the proton is being removed from this position, attaining a charge of 0, attaining a net charge of 0. Further comes the turn of this amino group, further comes the turn of this amino group, and hence it attains a charge of -1. So whenever it comes to histidine, this was the Zwitter ion, and how we will calculate the uh, pi, we will be calculating the pi, we will be considering the pk just immediate to the 0, to the pi, to the neutral charge, pkr. What was pkr? Pkr was 6.0 plus, what was the pk2? Pk2 was 9.17 divided by 2. Again, let's do the calculation. It's 7 1.9615 divided by 2. So pi comes out to be 2 sevens are 14, 7.59. Here's the pi. So this is the value of pi. So this is how we can calculate the isoelectric pH. So what conclusion do we derive from these titration of amino acids? So we clearly need to understand that addition of either H plus or OH ion to a Zwitter ion, it will result either in a net positive charge or in a net negative charge, as we've seen already. So depending upon the pH of aqueous solution, amino acid Also, the protein, they can exist in different ionic forms, and this property of amino acid, it is exploited. And also the property of amino acid implies to the proteins, because proteins are composed of amino acid, so they can behave as a weak acid due to the COOH group and weak bases due to the NH2 group. So it is employed effectively in nature because these molecules, now see why we are understanding all these, why we are studying the titration, because these molecules, they are acting as a buffers in the body fluid. So one of the important functions served by the proteins in the blood is to maintain the pH of the blood, which is within the narrow range of 7.35 to 7.45. So that's all for today. In case of any queries, please let me know in the comments section. Thank you so much. Stay tuned.